So this lesson is on karyotypes. And a karyotype is just an organized profile of a person's chromosomes. And it doesn't just have to be a person, it can be any living organism. This one happens to be a human karyotype. And I can tell that by the fact that there's 23 pairs of chromosomes. And you'll notice that the chromosomes are arranged by the length of the chromosome. So number 1 being the longest, and then they decrease in size. And the 23rd pair actually doesn't have a number. It has an X and a Y. So these first 22 pairs are called autosomes, and the last pair are called sex chromosomes. So when a scientist looks at a cell when it's dividing in mitosis and they see the chromosomes, they are not organized this way. But what a scientist can do is they can take a picture of chromosomes when the cell is developing, and then they can reorganize them with their matching pair, their homologous pair, and they can observe them using their size, their banding pattern, wherever their centromere is. And they can tell information about whether all of the pieces of the chromosomes are there. And the other thing that they will look for is if there are too many or too few chromosomes or pieces of chromosomes missing. To get even more detailed information, after 2003, when the Human Genome Project was completed, now scientists can compare and locate to see if entire genes are missing, but they would have to do further testing. So this is some information about how somebody could have too many or too few chromosomes, and this goes back to meiosis. If in meiosis, chromosomes fail to separate either during meiosis 1 or meiosis 2, when metaphase, when the chromosomes typically should separate. So here in meiosis 1, the tetrad, those homologous pairs, should be pulling apart. If they don't, it could result in one of them having too many chromosomes and another one having too few. And then you can see the gametes are going to have either too many or too few. If the mistake happens in meiosis 2, when the duplicated chromatids are separating, then you could have a result where one of the gametes has too many, one has too few, and the other ones, though, could be normal. So there really isn't a way to determine when the non-disjunction occurred, but it's just possible that it happens in both of those steps. And if this is an error that's occurring, notice none of the gametes are going to have the right number. But if it happens in the second phase of meiosis, some of the gametes could be normal. So chromosomal non-disjunction in humans, it could result in either the male or the female having an abnormal number of chromosomes, either too many or too few. And then the offspring having too many chromosomes would be called trisomy, and if having too few is monosomy. So there are different disorders of trisomy and monosomy, and I encourage you to kind of look those up to learn more information about those. One last point, that as we talk about karyotypes in genes, some of our obvious human traits are really impossible to associate with just a single gene or just a single chromosome. Scientists are learning more that it's multiple genes, multiple chromosomes that can control some of our genes. So like our eyes, our ear shape, height, and skin color, those are controlled by many genes. We also have to remember that a lot of our personal traits are only partly governed by genetics, but they could be influenced by our environmental factors. So height, weight, and personality can be influenced a lot by what you do or how you live your life. You could weight, when we talk about metabolism, and there's a lot of different theories out there that say that, this is how you're destined to be. But if you think about it, if you ate McDonald's every day or you ate a different type of food every day, we've talked that those things can alter your genetics. And that's actually a field of genetics called epigenetics that's becoming, this is the next step of where scientists are trying to understand how the environment might impact our genes. And even some traits are influenced by external environment like temperature, light, and pH. Example, the Arctic fox, its fur color changes due to gene expression that's impacted by temperature changes. So you think about those are adaptations that also help organisms survive. So as we venture into genetics and our topics on evolution, these types of things are something to always keep in the back of your mind, that we don't have all the answers yet.